All right, well, thank you for attending today's research translation seminar. I'm going to talk to you today a little bit about my own journey developing a new drug. And my partner here today is Brian Dymock. He is the Director of Chemistry at QEDDI, which is the Queensland Emory Drug Development Initiative. Discovery Initiative. Okay, so I'm going to talk to you a bit about the clinical question, observations from the clinic, the models that we developed, and how that led me to settle upon a target. And Brian will tell you about what QEDDI does and show you some of the data from the drug. So I probably don't need to introduce to you the concept of organ transplantation, but unfortunately, even today, many patients who need a transplant don't get one. And those that do are very fortunate. But their life looks very different from the point of their transplant. They must take anti-rejection drugs daily for the rest of their lives. They take cocktails of drugs such as glucocorticoids, calcineurin inhibitors, and inhibitors of nucleotide synthesis. And I've highlighted tacrolimus in red. This is the drug that the vast majority of these patients take. The clinical problem for these patients is that the mechanisms that have been suppressed to prevent graft rejection are the same mechanisms that the body would use to fight against cancer. So these patients have a very significantly increased risk of developing skin cancer, squamous cell carcinoma. The tumors are more common, they're very aggressive, and they're often in high-risk locations such as the face. These patients have to have these lesions cut out to prevent metastatic spread, and some patients here at the PA hospital will have 10 excisions a month. So as you might imagine, SCC is a leading cause of mortality in transplant patients. So some observations from the clinic. People have looked at removing immunosuppression from patients, which does result in skin cancer regression, but also results in transplant rejection. And as you might imagine, maintaining transplant health is paramount. They've also looked at reducing the dose of immunosuppression. And in these cases, again, you see a reduced cancer incidence, but an increase in transplant rejection. The take-home message here really is that there's a very tight association between the level of immunosuppression or the functionality of the immune system and the ability of the immune system to fight back against SCC. Interestingly, not all of the drugs that the patients take confer the same risk. So today I'm focusing on two. One is tacrolimus, which is a calcineurin inhibitor, and it prevents T cells from becoming activated by interrupting the pathway downstream of the T cell receptor. The other drug is rapamycin. It's an mTOR inhibitor, and rapamycin will allow T cells to become activated, but then starves them of those signals such as IL-2, which they need to proliferate and differentiate to become effector cells. But the two drugs essentially prevent T cell function. Now, some years ago, it was published that if you take some patients who are on calcineurin inhibitors and you convert them or switch them onto a medication regimen, which includes rapamycin, so you keep all of their other drugs the same. Just doing this reduces the risk of new SCC formation. Importantly, patients still get SCC, but they get fewer SCC. But also interestingly, you see regression of pre-malignant lesions in some patients. So you might imagine that we should switch everybody to rapamycin, but rapamycin itself has a high degree of drug discontinuation because it has a lot of severe side effects, such as an inability to heal wounds. So some years ago, I established models in the laboratory to try and understand why these two drugs are acting or how they have a different outcome for SCC formation in the patients and to try and uncover those mechanisms. So we developed a UV-induced model of SCC. It's a regression model, which means it can be controlled by a healthy immune system, and I'll show you that in the next slide. And we also developed diets which would allow us to deliver the drugs orally, which is how the patients take them over the long term, whilst also achieving clinically relevant target thresholds in the blood. So what you're looking at here is immune competent FVB mice. And if you put them onto a tacrolimus diet and inject tumor cells into the ear, you can see that the tumor cells gradually grow and form a tumor over time. If you remove the tacrolimus, in this particular case, it was day 13, and we simply take away the drug diet and put them back onto a healthy diet, you'll see that the tumors continue to grow for a few days. And I believe this is because it takes time for tacrolimus to be flushed out of the system and for the T cells to become active and go to the tumor. But after that, you can now see on the bottom of the slide a fairly rapid regression of the tumor. And out by day 55 or so, there's very little evidence that there ever was a tumor. So using this model in many different ways, we've been able to establish the mechanisms of tumor rejection. And so what we often do is at the day when we remove tacrolimus here in the red box, we'll inject the mice with antibodies to deplete specific immune subsets. Now we've done that for many different immune subsets, but it's really the depletion of CD8 T cells shown here in red that determines whether or not a mouse is able to reject its tumor. So if you compare the red line, which is CD8 T cells, with the blue line, where the mice are rejecting their tumor, that's CD4 cells, you can see the difference. So CD8 cells play a very key role in driving tumor regression. If you deplete both populations, that's the gold line on the left, you can see that the tumors grow faster. So I'm not saying that CD4 cells don't play a role in controlling tumor growth, but they're not the immune cell that determines whether or not tumors will regress or not. So going back to our two drugs, I've already sort of let the cat out of the bag a little bit and mentioned that tacrolimus will allow the tumors to grow in an FVB mouse or an immune competent mouse. But if you put the mice onto rapamycin, you can see that the tumors don't form on the graph at the top right. And the graph on the bottom right shows us that if you establish tumors in mice by feeding them the tacrolimus, and then you switch the diet over to rapamycin, now you can result in tumor regression. And just a note for later, you can always see in the control, if we remove suppression entirely, we always see a much rapid rate of tumor regression in the mice. Interestingly, if you do similar experiments in rag mice, which lack T cells, you see slightly different results. For example, because rag mice don't have T cells, they can't control tumor growth. If you put them onto tacrolimus, they also can't control tumor growth. But again, when we have rapamycin, they can control the tumor growth, at least in the early stages of the experiment. And this tells us that the ability of rapamycin to prevent new tumors forming possibly has little to do with the adaptive immune system. But the interesting observation for me is this experiment at the bottom, where we allow tumors to become established in rag mice. And these mice were all injected with the same tumor line. It's just that some cells grow faster than others. And so I've grouped them into fast-growing tumors and slower tumors. And you can see in these mice, in the absence of T cells, rapamycin is not able to prevent tumor growth. It has an effect on the rate of tumor growth, but the tumors do not regress. And for me, this was quite a turning point in my thought process, because it made me think that rapamycin is probably allowing residual T cell function. And indeed, we did a study of that to determine whether our drug diets were creating a low-dose rapamycin environment in the skin, which promotes memory T cell function. We know from the literature from other mouse models that low-dose rapamycin promotes the differentiation of memory cell precursors during the activation stage. So this essentially tells us that the rapamycin's effects in patients are likely to be T cell dependent. And this creates us with something of a problem, because patients take tacrolimus. Tacrolimus prevents T cell activation. And so you can imagine if you combine the two drugs, you won't see the beneficial effects of rapamycin, at least on tumor regression, simply because tacrolimus is preventing those T cells becoming active in the first place. But while developing these models, it was quite obvious again and again from experiment to experiment that just taking away tacrolimus always caused this very rapid regression of the tumors in all of the mice. And that made me start to think, well, is there a way we can do that in patients? And remember, we can't just take away tacrolimus entirely from patients because their transplants will reject. So the idea that I came up with was whether or not we could just locally control the amount of or the effects of tacrolimus in the skin. So this diagram on the right is a little misleading. I'm not suggesting that we allow the immune cells all over the skin, all over the body to become active, but simply allow for periods of time, transient periods of time, T cells to become active in areas where we know that there's a high SCC burden. So in 2016, I approached QEDDI and I asked them whether they could help me to develop a compound or a drug which could be used to disrupt the effects of tacrolimus in the skin only. And Brian's going to take over, tell you a little bit more about QEDDI and how they did that.
Great, thank you, James. So I'd like to tell you a little bit about QEDDI, what it is, what it does, why it's here, and give you some information on the studies that we did with James and Maggie, who's in the audience there. So QEDDI is actually a partner for any UQ academic group, specifically focused on small molecule drug discovery. So we don't do peptides, we don't do proteins or other biologics, focused on small molecules. And the principal objective is to take your innovative biology and translate it through the drug discovery process to come out with a product, preferably a drug molecule that we can then develop with an external partner, such as a pharmaceutical company or a biotech company, and eventually get that into the clinic where we can test it in real patients and add value. And we think that's a very exciting prospect, working together in a multidisciplinary team to get real value. So as you probably know, the therapeutic process starts off with biological research in the early days, which is mostly what you guys are focused on. Then there's a little gap here where we are doing some translational research, in other words, discovery of the drug, development, optimization of the drug, and development of the drug ready for clinical testing. Then into the clinic for a number of years, fairly expensive process, then regulatory review and approval and availability for patients. So this is a fairly long process, which could take up to 12 years, the development stage, and the research stage could be anywhere, we would estimate typically 4 to 6 years. So it is a fair amount of time. And I think you saw James mention there, September 2016. So we're now into 2021. So we're right in that zone for this project. So I think these are fairly reliable timelines. So this gap, which I mentioned, is actually got a lot of challenges in it, often known as the Valley of Death. And you can imagine if you're crossing on this rickety old bridge, that just shows the nature of the risks here. There's many, many things we need to think about when we're doing target-based drug discovery. Is the target druggable? That just means, can you develop a small molecule that would fit into a discrete receptor that would be potent enough to exert a pharmacological effect at the site of action? One of the biggest challenges is getting a drug to the site of action. So we've got chemistry. One of the big things QEDDI has is chemistry. We have 10 industry experienced chemists and we outsource a bunch of chemistry as well. QEDDI in total is about 20 people. So how you progress a screening hit. So if you do have a screen and you screen a library of compounds, how on earth do you take that forward? So that's something we can help with. And other aspects are listed here, which you can read. Importantly, safety consequences of the target. So we spend a lot of time worrying about safety. So if we inhibit this target in a patient, what are the likely side effects going to be? A very important aspect. And of course, early on, we worry about target validation. Is the target actually, if we inhibit it or stimulate it, is that actually relevant in the disease? Then on to commercial aspects. So UniQuest is the parent company of QEDDI, and UniQuest is focused on getting commercial results from research projects. So they are there to help you to file patents, to get value from your project in the long term, value for you and for UQ. So questions around the market. Is there a market for the idea? And I think James showed you that the market is very clear and very specific in the case of SCC and organ transplant recipients. Patentability. This is an area UniQuest are very strong on. How do you write a patent? When do you write it? How do you go about filing it? How do you choose a patent attorney and so on? What does pharma want to see? So these are our customers. These are the guys with huge amounts of money. They just can't wait to get their hands on an exciting project to invest in it for clinical development. So what do they want? And are we barking up the right tree, or are we barking up the wrong tree? Choosing animal models, which James shared with you, can be an important and difficult aspect. Also regulatory, this feeds into the clinical aspect. What do regulators need to see? And this can really slow you down if you get it wrong. All of that stuff, and last but not least, manufacturing. So manufacturing means scaling up, making more to the required standard that you need for that stage in development. So lots of different needs to support translation, from discovery through to worrying about safety and commerciality. And that gap there is what QEDDI fills. So QEDDI was modeled on successful models. There's over 100 so-called academic drug discovery groups in the world. And so we learned from that. One of the key things is to link your group to a commercialization group. So QEDDI being a part of UniQuest is actually extremely handy, because these guys are full of commercialization expertise. Very defined scope. So we don't just work on anything, as I said, small molecules. We work on identifying hit molecules, and then we optimize those into final drug candidates. And we can also take them into preclinical development, but we prefer to nominate the candidate and then outsource that to a pharma company if they're willing to pay for it. So that would be a licensing process. And I'll say something more about commercial outcomes later. And of course, the key thing is it has to be commercially relevant. So if it's something that's well treated already, there's not going to be a demand for that. So we're looking for those areas of high unmet medical need. And the facility ourselves, we have medicinal chemistry and computational chemistry, as well as a group of very competent and experienced biologists. And we can also do screening of libraries, relatively small screens of maybe 10 to 30,000. And one of the key differentiators is people in QEDDI have industry experience. So they've seen it and done it, and in many cases got the T-shirt as well. So we know what it needs to look like. And a very specific remit, so working with academic groups in UQ. So every QEDDI project has a UQ collaborator who is invested in the project and works with us on the project. So for commercialization, we start off with discussions. Like James mentioned, he approached QEDDI, and there's a selection criteria for projects. And the project, once it's established, would have the UQ researcher bringing their unique and innovative insight, and QEDDI bringing its drug discovery and commercial experience. Then finally, we have commercial outcomes. And we're at that stage now for a number of projects in QEDDI, including James's project, which is very exciting for us. We're going out there talking to investors and looking to bring in further, either a licensing deal or further investment to create a spin-out company, which can then be a vehicle for developing that drug as an actual clinical therapy. So in the case of James, he approached this with his project on squamous cell carcinoma, which was a really interesting project for us. Slightly unusual in that it's not your traditional oral small molecule. It's going to be a topical on the skin. So looking to combat the effects of tacrolimus in the skin without affecting the patient's graft. Very important indeed. So what we needed here was compounds to validate the concept that James had, and also developing these compounds to have the right drug-like properties for topical delivery. Of course, we're all familiar with topical drugs, a cream or a lotion or an ointment, but actually making that work and understanding it is a very different ballgame. So it's not as straightforward as you might think. And modeling the clinical disease preclinically is also not straightforward. So we had very clear objectives here. We want to be in the skin, but we don't want to be in the system. So no systemic exposure, because that would threaten the graft. So reversing the effects of tacrolimus. So what we decided to do is to look for molecules that could compete with tacrolimus for its binding site on FKBP12, which when tacrolimus binds there, that then forms a ternary complex with calcineurin and has the immunosuppressive effects. So at the top here, we have tacrolimus, so otherwise known as FK506. This is an IC50 curve. We always like to operate using a dose response curve. So we can see here that the KDE is around about 1 nanomolar. And We looked at a number of compounds in the literature. We synthesized them. We tested them in an assay, which this is a FRET assay that was outsourced to a company in the UK. So we have a wide network of companies that we use for outsourcing. It's easier and quicker for us to do that than to bring the assay and develop it in-house, because we thought we're not going to use it for a vast number of compounds. And these guys already have it running. So we paid them to do this for us. It confirmed the right result for tacrolimus. And for 2361, which was our candidate compound, after looking at a range of other compounds, we found this compound had 2.8 nanomolar IC50. So we were satisfied with that. It's a very strongly potent compound. And we felt that had a good chance of displacing tacrolimus from its active site and freeing up the immune system in the skin, as long as 2361, our drug, doesn't get into the systemic circulation. Because this is just the isolated target. We moved on then to look, working with Maggie and James, in T cells, and mouse and human T cells. So this mouse T cell assay, you can see there, when you have tacrolimus present, you have a smaller bar here. When you start adding in the drug, you have an increasing size of the bar, which indicates that the drug 2361 is displacing tacrolimus from its active site. And we can see here that there's also no toxicity. And similar in human cells, so using human cells for the greatest translational applicability, and mouse cells because that's the model that we're working with. We take that forward into the model. So James described his model. So this is the tacrolimus treated vehicle group, and you can see the tumours growing quite substantially there, up to nearly a cubic centimetre. And the drug treated group, so we started the drug treatment here at day 8. So once the tumours are established, this is intratumoral injections. It's not ideal, but it is the best way of modelling this because it's a subcutaneous tumour, so a topical administration wouldn't work. This was just proof of concept though, but it was more than good enough for us to get excited. So you can see when you do daily injections, after a number of days, around about 12 days, you start to see a differentiation between the treated group in orange and the vehicle group. So we were very happy to see this tumour slowing down its growth there and actually stopping growing. And we were happy that that was really what we wanted to see. But of course, adding more mechanistic information is always good, and that would take a much longer talk to tell you about. But importantly, in terms of drug distribution, we wanted to show that the drug was not getting into the system, as I mentioned before. So this just shows, we did a study with mouse skin and the mouse ears, and we showed that there was a large quantity of drug in the skin, and it remained in the skin up to 24 hours, in fact, probably for several days. And in the system of the mouse, in the blood system, we didn't see any drug. So we're very happy with that. So that ticks the box for potency, mouse cell, human cell, animal model, and drug distribution. Just a few of the things we like to look for. So as a team, we were very excited about this. This has been taken on now. Further mechanistic work has been done to confirm the mechanism of action via CD8 T cells. And we are taking it forward, looking for investment to take it into preclinical development. So for potential outcomes from a QEDDI project, well, a package of data, preclinical data. Some of that data may be using human cells or even primary cells from humans to validate the mechanism. One outcome, perhaps the outcome that we would desire initially, is a direct licensing agreement where the product is developed in the company. So this could be GSK, Merck, Pfizer, or a medium-sized or lesser well-known pharma or biotech company. All have a great deal of money there for projects that are attractive and have commercial possibility. So we have to convince them on the science, and we have to convince them on the commerciality of the project. So that deal would be discussed, the deal would be made. This is what UniQuest does. It's their bread and butter. And there would be milestones potentially agreed, or even downstream royalties are possible for the three parties here, UniQuest, UQ, and the academic researcher benefits commercially from this arrangement. Similarly, outcome 2, it's a different way of doing this, is to bring further investment. We're talking millions of dollars here. It's not small amounts of money, say at least 5 million to get from where we are now into the clinic. And this would be used to start a spin-out company, a new company. And that would be a vehicle to manage those funds and to develop the product to get a return on investment, potentially later selling the company or licensing the product to realize that return on investment. So that would be a very exciting stage to get to. And QEDDI is now at that stage in its growth. It's about 5.5 years old now, where it's looking to get these outcomes, and that will validate the model. And hopefully QEDDI will then be able to continue on longer into the future. Initially, QEDDI's funding is for approximately 10 years. So that's quite a good runway to try and realize some of these objectives. So working with QEDDI is fairly straightforward. It's very collaborative, very sharing. We have joint project meetings. We have a lot of interaction as appropriate on the science to get the projects moving. And also, one of the values for being in UQ is to access suitable translational models, potentially available at UQ, perhaps with another collaborator. But typically, that would just be a separate arrangement if we needed to access a different model. And collaboration around patient samples is very important as well. This is key for target validation. If you can confirm the target is a relevant target in human material, that's a lot more relevant than in a preclinical species. Also, we can provide for you advice on small molecule drug discovery. So if you find that your project is not suitable initially, we're happy to advise you and support you in your endeavours to progress the project. And this could be in any areas of drug discovery research, including library access. We're members of the Australian Lead ID Consortium, which has the Australian Drug Discovery Library. And that's the team, just a few of the people there you might have seen over the years. Mark, who is ultimately running QEDDI from his position in UniQuest, and Andrew, who's our Director for Partnerships. He's given a number of talks here in the past. You may have seen him before. And Craig, who's our Senior Director for Life Sciences. So it's Craig who would do the actual deals there, and a whole load of industry experience. So I think I've probably ran out of time now, and just some acknowledgements there. The people who actually do the work. And yeah, we have a few other things here on project selection, but that's the sort of thing we can talk to you about later if you have a specific interest on your project. So thanks very much. I think we're happy to take any questions if anyone would like to ask one now. Yes. Yeah. Yeah, so the question was, once the project is accepted, how much back and forth collaboration would there be? And actually, I would say in the project with James and Maggie, this is an excellent example of extensive collaboration at a very deep and detailed scientific level, all the way down to actively running an animal study and making tweaks and improvements as you're going along within that study, even on a daily basis, would you say, James, that happened there? And really very detailed. Sometimes you may not have much discussion for a couple of months, but when things are really moving, it could be daily contact. So whatever's required. Yeah. Yeah, so yeah, you're right. In this case, James had the model, so it was perhaps faster and I wouldn't say anything's easy in this business, but faster and easier to access and tweak and optimize it. You have much more control there. But when the model is not readily available, which is actually more likely the case, as we found from our other projects, we would then be looking for the model in a CRO. We prefer a CRO, a contract research organization who has the model established, or another academic group. It could be anywhere, preferably in Australia, where we would say to them, hey, look, we're interested in testing our compound in your model. Would you be willing to run that model for us for a fee-for-service arrangement? So they would not be brought into the collaboration. They would not know anything much about the compound. They would just run the model, and we would pay them for that. Mm. Yeah. Absolutely. Excellent. Yeah. Yeah. Actually, that would be such a great resource to actually, just for people listening, the comment there was to have a database of available animal models or other models, perhaps even cellular models, that people could access and have that database there. That would be extremely useful, because we've actually identified that, I think, as a key requirement in a project that is not well met right now. So instead of running off to a CRO and spending an absolute fortune on getting perhaps even a suboptimal study, why not do it with a real person you can meet with here in the country? It doesn't need to be in the same institution. It could be in another institution, preferably in UQ, always easier to collaborate, but could make it work elsewhere. I mean, another example of this is in the drug screening area, where the National Drug Discovery Centre in Weehai are screening full deck screens of 300,000 compounds for people. Now, they can either do that as part of their application process, or you could pay them to do it or collaborate with them in many ways. But people don't necessarily know about it. It is there, but do we know? Yeah, so Griffith actually have Compounds Australia, which houses the library, and they also have a number of other compounds people can access. And now there's this Australian Drug Discovery Library, as it's called. It's been quite heavily funded, and it's got 330,000 high quality compounds. Compounds Australia now have these, and this is accessible to all researchers in the country. And UniQuest are a partner in this, and we've helped develop the library. So if you have any questions about anything relating to screening, drug discovery, translation, animal models, whatever, do contact us and we can help you. More than delighted to do that. Really easy, and we're so pleased to hear from people, as we can definitely help you with anything on chemistry. That's my comfort zone, but many other things as well. So do please contact us. No strings. Happy to have a coffee and share what we have and try and help you. Thanks for the comments. Yeah. Any other? Yeah. Yes, it's a good question. At the moment, yes, but we're working on it. So the question was, if you're at QUT or another non-UQ institution, do you need to have a UQ partner? The answer is yes at the moment. UniQuest is saying that it wants to work with UQ researchers at the moment. It's only allowed to for this initiative, given the nature of the funding, etc. But I think the management are working on a way to expand that, just to try and access other promising projects, really. But at the moment, it is UQ focused. But if you do have some knowledge of a good project or someone who would like to do something, I would really like to hear about it, because that could be the way, a vehicle to get this thing done, so we can do projects in the let's just say in the wider Queensland area to start with. And I think at the end of the day, we'd probably still want to have a UQ collaborator in there somewhere, because we're doing this for UQ, for the UQ organisation and the researchers within it. But that's a good question. It's a complicated question, though. But thanks for asking.
At this time, we're not, and that's simply because of those studies that have looked at swapping out tacrolimus specifically and the effect that that has on SCC. You're quite right, we might get halfway there with our approach. We may need to think about whether those patients, for example, are given different steroids, or these are really clinical questions. But yeah, I guess it's a journey that we've just started on here.
Great. Thanks.
Well,
Thanks,
thank you,
everyone. Thanks for coming.